And now our second talk for the Control, Protect, Cure research theme, Jochem Vink, tēnā koe. Why understanding the biology of Phytophthora agathidicida matters. Jochem is a postdoctoral research fellow at Victoria University, working in the group of Mycelial Growth for about a year, studying the mechanism of infection and spread. He has completed his PhD at TU Delft in the Netherlands, where he's originally from, and has worked on biological research projects in Peru and the United States. Jochem first encountered Kauri on a trip to Northland 5 years ago and still feels humbled by that experience. Tēnā koe, piki mai.
Kia ora, Stacey, and tēnā koutou. So, ko Jochem tōku ingoa. I'm a postdoctoral fellow in the Mycelial Growth group at Victoria University in Wellington. And first I'd like to thank the organizers for this great hui and just the awesome corridor that we've had yesterday and this morning. It's been very inspiring. Today I'd like to talk about Phytophthora agathidicida, the biology of this organism, and why this might matter to how we manage this organism. So my background, I'm from the other side of the world, where I studied kind of fundamental biology and chemistry, but I was missing something, and that thing was that I wanted to be involved with nature conservation. And seeing these trees on my first trip 4 or 5 years ago, that was really a life-changing experience for me. And I'm so blessed and humbled that I'm able to work on these on this project at the moment. So what I study is this person, and I'm very glad Marion already given kind of an introduction to PA. So it has a very complex life cycle. It spends part of its life in the roots of Kauri trees, but then in the soil it can spread into different kind of spores. So Marion already talked about the Zoospores that are swimming around in the soil, but there's also the Oospores, and I will talk about them later as well. And so we're kind of asking ourselves, can we understand the mechanism of how is it going through this life cycle? So how is this whole thing regulated? And the goal of that is to improve the robustness and reproducibility of the work we do in the lab, but also to identify perhaps conditions that might help or reduce the disease in the nahiri, and to find potentially unique weak spots that we can target. So mycelial growth in a petri dish looks like this. It's a bit hairy, fungus-like growth, but it's not a fungus, it's a different type of organism. And in plant roots, this is what it also looks like. But at some point it decides to go and do something else. It decides to go and make spores. And we found that usually depends, when that happens is, it depends on the nutrition. So it's happily eating food from its environment. At some point it runs out of food, and then it decides, I'm going to make spores to look elsewhere. And it can do two different things. It can make the Zoospores, which will swim around, and it can produce the Oospores, which will stay put and just survive. And what we did is we just made a very simple composition of media, just of the very basic ingredients that it needs. And by changing the ratio of these components, we were able to make media that either only produces these Zoospores or only produces Oospores. So we have now a better understanding of what is driving these different paths. And the bonus of it is that we're now able to produce them more reliably and with higher yield. So we're able to better study them in the lab. So the Zoospores, so this is already kind of a video that Marion showed before. So they get released from this tissue, in this ball here, and they're the key route of dispersal. And as Marion also mentioned, they can smell the Kauri trees, and this is a process we call chemotaxis. And so this swimming, just to give you kind of an idea, probably in the soil, it probably is about a meter distance max that it would swim. So most of the dispersal goes via other pathways, but when it's close to a root, then to actually get to the root, it will use that kind of smelling to get to that last bit. So we have had a lot of tools in the lab to study what attracts these spores, and we've been able to identify some of those things. But actually, we weren't able to figure out what repels them. And this is another kind of thing that we wanted to know, because maybe could be something that could be useful in the field. Right? If we know what repels them, maybe we can scare away the zoospores. And so now I developed some tools that we can use. So we're basically just directly imaging them on the microscope and then tracking them with machine learning algorithms to see where they're going. And in this way, we can actually also figure out what is repelling them. And we're testing all kinds of compounds ranging from plant extracts, microbes, but also just synthetic chemicals. Then the other one is spore type. So the Oospores are a very different bunch. So they are very tough. They don't move at all, and they will stay in the ground for years. And so they are the spores that are made for survival. And so when we're thinking about what do we need to know about these spores, is we need to figure out a way to deal with these, because they will be the one that will be on your shoe when you're walking around in the forest. They will be the ones that are in nurseries. So how can we deal with those? And we've developed an assay that we can test reproducibly and on a large scale all kind of treatments and the effect on those treatments to the Oospores. And so we get kind of a traffic light system where the Oospore will turn green if it's viable, red if it's not, and orange if it's damaged. And so we can kind of test hundreds of thousands of different things and see, okay, what are the things that trigger these Oospores, and maybe find for particular applications, particular treatments. So again, it's going to look like on a larger scale. Now, how does this whole translate, this microscopic world translate into the larger scale, the Ngā Rākau and the So what I think is that we're all carrying little pieces of the puzzle, and the forest is just this amazingly complex thing, and we cannot ever get the forest into the lab and study all of its complex interactions. So what we can bring to the table is these very reductionist, very simplistic pieces of the puzzle, looking at the microscopy. But then there's this other approach, and this is also very exciting for us, this Mātauranga Māori approach, where you look in a holistic way at the forest. And talking together and having this kind of feedback loop, we can generate ideas back and forth and exchange ideas, and then we can start to target things. And so one example is, for example, so here we have some opportunities and risks that might be involved in the forest, and so plants that are associated with Kauri could be an opportunity, but also could be a risk. And so what we are doing now is going in field studies in collaboration with Te Roroa, and they are planting certain plants amongst Kauri trees and seeing, and because they know which plants are the most effective ones, because there's thousands of species and we can't test them all, but they have the knowledge to think these are probably the most probable ones to have an effect. And we can test that, and we can go back to the lab and look at all these effects on both the Oospores and also on the Zoospores, and by knowing that, then we can be, and then we can go back to the field trial and be like, okay, probably a combination of these would help. And so this is ongoing, and this is a very exciting space for us to work in. So just as a takeaway, I think understanding the biology of Phytophthora matters a lot, because it can increase our ability to further study this disease, but it also provides a chance to develop more specific and effective management tools. So with that, I'd like to thank my supervisor, Jochem Vink, and the entire MME lab. So I'd like to thank Ngā Rākau Taketake for giving me the opportunity to be here today, and for funding, I thank the Netherlands Organization of Scientific Research. I'm happy to take any questions and also suggestions now or after. Yeah.